Hi everyone, welcome back to the channel, and today we will be understanding the process of DNA underwinding. If you have seen my previous video, and in that particular video what we talked about is the DNA supercoiling. Now in this video we'll try to understand what is the connection between DNA underwinding and DNA supercoiling. We'll try to understand some of the important and basic facts and concepts regarding these processes and how they are important as far as the typical DNA metabolism is concerned. So as you can see, let's see what we have today. So as you can see, I have a few points written. The first one is what is DNA underwinding, right? So that is what we need to understand. DNA underwinding is nothing but it's a condition when you have few helical turns in the DNA molecule. Now if I simplify this, I'll take the example of the DNA turn. We have understood the basic structure of the DNA, and we all know, all biology students at this particular point, they all know what is the structure of DNA molecule. It's something like this. So let me draw it. It's something like this. It's a double-stranded DNA molecule, so that is what I'm making, right? So it will start like this, and the second strand will run like this. And as you can see, you have turns in this, and if the typical number of turns per few nanometer of the distance, they are changed, and the turn that you are getting in the new DNA molecule, say it may be less than the 3 that we have. Because of that, what will happen? You will have a strain, and it will be a little bit stretched, isn't it? So as you can see, it is more like a stretched DNA molecule. So because you have fewer helical turns, that will cause DNA to stretch, and because of that strain will be there. So DNA underwinding is important for many things. You can see the first one that I'm mentioning here is the closed circular DNA. You need to understand these conformations. So what is closed circular DNA? Closed circular DNA, let me use another color for that. Closed circular DNA is nothing but the molecule when the DNA molecule when it don't have any breaks. So it is like a closed circular DNA. Next, whenever the DNA is isolated, a particular cell or particular organism, it will have a characteristic pattern of supercoiling. That is another point. And because of that, its conformation is going to be different from different sources. For example, if you are taking the DNA from the virus, and as in this case you can see bacteriophage, its DNA is going to be a little bit different than the DNA of the bacteria, right? So on the right side you're seeing bacteria, and the supercoiling process. And then if you talk about the plasmid, just the plasmid, which is the extracellular piece of a DNA molecule present in the bacteria, its pattern is going to be different than the genomic. So depending upon the source, the supercoiling will be different, and it will be typical. It will be characteristic of that particular source. Now these are the two basic things. And now why underwinding? Underwinding is important because whenever there is a DNA underwinding, strain will be there, and that will cause DNA to supercoil, or it can also cause the DNA strand to separate, and that is important for various cellular processes. So I hope this is clear that we talk about DNA underwinding process because it is important during various cellular processes. DNA needs to be separated. The strands they need to be separated, and various enzymes they are working for that particular process. And then we also talk about the closed circular DNA, where DNA is relaxed, and because of that underwinding process you can have the conformations like supercoiled conformations. So they are connected. Now let's move on to the second part, and let's see what we have in there. All right, so now we are in the second part, and as you can see, whenever the DNA is strained, so here What I'm trying to mention is now DNA is strained. Maybe DNA underwent the unwinding process where cellular machinery is stretching up the DNA, or some other forces they are causing the DNA strand to stretch. What will happen? Two possibilities are there. One possibility is it will lead to the supercoiling of the DNA, and that is what I'm trying to show you, that this is the closed circular DNA, and now it is being supercoiled because of that, the strain that is there in the DNA because of the DNA underwinding process, right? And the second thing that can happen is the strand separation. Strand can also get separated. So as you can see, this is highly important. So if you get this particular message, and I'm sure this is going to help you understand in a lot of important cellular processes. So what else can happen? Second is, in case of the genomic DNA, probably what will happen is the DNA strand, they can also get separated out, and then the enzymatic machinery can access this particular space that is getting generated. Now what will happen in this case? You can clearly see in case of in vivo, that means inside the organism, inside the cell, you have huge DNA. It's a coil. It's not in the supercoiled, specifically in that supercoiled form, which is for the plasmid. It is different than the plasmid, and then what will happen in that case? It's not going to undergo in that particular process, but what will happen in that case is it's going to lead to the strand separation, not into the supercoiling. If there is a DNA strand underwinding. So underwinding in case of DNA can lead to strand separation. That is the first aspect here. It can also lead to the supercoiling. Now, in this case, what happens is, let's say we talk about the plasmids. In case of plasmid, they are independent, they are circular, they are smaller molecule. What will happen? They will immediately acquire this closed circular DNA conformation. Why? Because it is, for them, it's less energy consuming process, correct? Because in this case they don't have to consume the energy to break the hydrogen bonds between the bases, so it's more convenient to get these conformations in case of plasmids. Next, if we talk about DNA in vivo, what will happen? This is most likely not going to happen if there is a DNA underwinding. What will happen in that case is going to be the strand separation, because that is going to facilitate the access to the information. So as you can see, especially in case of the transcription, where enzymes, they need to access these bases so that the complementary RNA will be synthesized from the DNA. So there, this particular process is highly important. So as you can see, the forces, how the forces are acting, and how in one aspect you get the supercoiled DNA, in other aspect you get the strand separation. And then we have discussed the significance of both of these processes. All right, so now let's move on to the third part. All right, so now we are in the third part, where I'm going to extend whatever we have discussed in little bit more detailed way, where as you can see, I mentioned DNA underwinding as DU. In the cell, when the DNA underwinding is happening, various enzymes are involved. Various enzymes are involved in that particular process, and there is a significance of that process. I already told you the information need to be accessed, so that is why DNA underwinding is important. Next is DNA underwinding can lead to supercoiling, that we already talked about. Second one is DNA metabolism. DNA metabolism is another part where enzymes are involved to access the information. We have already talked about that one, because whenever DNA underwinding is happening, you can have supercoiling. That is the first effect, and second one is the strand separation. So in case of DNA metabolism, when replication is happening, transcription is happening, you need to have that information accessed. Next, okay, as you can see here, in case of the DNA underwinding state, it needs to be maintained. Otherwise, what will happen? It will again revert back to the relaxed stage, which is the closed circular form. Correct? Which is basically not the closed circular form, which is the relaxed or circular form. Right? So you have two things here, which will keep the particular state of the DNA, which we are talking about, the DNA underwinding state. It needs to be maintained. And how it will be maintained? That either that particular DNA molecule need to change into a closed circular form. If the DNA is in the circular relaxed form, it will change its conformation to closed circular form, or it will be stabilized by the protein. If it is not stabilized by the protein, if it is not taking the closed circular form, maybe there is a nick in the DNA molecule. What will happen? Immediately, that strain is going to be again, help the molecule to go back to the relaxed state. So proteins are important in this case, as you can see. That is why there are a lot of proteins that are involved, especially in vivo processes. And second, in case of plasmid, that is why most of the plasmids, they are in the closed circular form, because they need to maintain that the DNA underwinding state. Alright, so I hope the topic is clear. I know this is very, very fundamental, where we talk about, again, some of the aspects of DNA supercoiling. We talk about DNA underwinding process, how they are connected, and what is the significance. These are some of the basic things, but they will help us to understand some of the advanced topics in the biochemistry, especially when we talk about gene and chromosomes. Alright, I'll come back with more information on this particular topic, and we have some interesting topics. After this, I will be talking about the linking numbers, which is again a very, very important topic in case of the DNA conformation or DNA metabolism. All right, please watch all the videos, and let me know if you have any questions.